Hello everyone, welcome back to the PolYneer's podcast.
In the previous episode, we mentioned our project is targeting c-MET and EGFR mutation. But how exactly are we doing? The answer is we are developing a novel bispecific antibodies.
Okay, here let me introduce bispecific antibodies first. Bispecific antibodies are a large family of molecules designed to recognize two different antigens. This property has been exploited to target two types of membrane receptors within a cell. In the case of NSCLC, these antibodies are designed to bind specific receptors on cancer cells and immune cells.
In this time, bispecific antibodies can act as a bridge between them. This can help enhance the immune system's ability to recognize and attack cancer cells more effectively. But this is the traditional bispecific antibody, which is an expensive treatment. In order to reduce the cost and increase the efficiency of the bispecific antibodies, we developed a new bispecific antibody to achieve this goal.
However, as the beginner of the research area, we still need to understand how to maintain the safety and the effectiveness in treating lung cancer. That's why our team reached out to one of the professors who is an expert in lung cancer research to shed some light on this. His name is Professor Tony Mock, who is an expert in biomarkers and targeted therapies on lung cancer. He is also a co-founder of the Lung Cancer Research Group and has led a number of multinational studies on personalized clusters of differentiation medicine in treating lung cancer. I believe not only us, but everyone could learn from him. Now, let's jump into a conversation between our curious students and Professor Mock.
Hello, Professor Mock. May you say hi to our audience, please?
Hello, everyone.
Thank you, Professor Mock. As we know, Professor Mock also dedicates clinical research to tackle the problem of NSCLC, which is similar to our project. Therefore, we would like to ask, how do you evaluate the safety and the efficacy of new drugs for lung cancer in preclinical and clinical setting?
All right, thank you. This is a broad question in terms of the new drug development for non-small cell lung cancer. But why don't we narrow a little bit to my understanding? You have developed a bispecific antibody, namely that target both the EGFR epidermal growth factor receptor and also with the MET. So this is a bispecific antibody similar to what is already in the market by a drug of amivantamab. So the question, in your drug development is that, how is it different from the existing drug? Are you a me-too drug, or me-better drug, or novel drug? Because your investigation pathway is different. Now, knowing that it is actually specifically inhibiting the EGFR and the MET, then you have to ask for the class effect toxicity, including like EGFR inhibition, which is actually EGFR is known to be existing in skin and mucosa. Then your skin toxicity as well as the GI toxicity has to be concerned. As compared for MET inhibition, then you have to worry about edema and weight gain and those kind of toxicity. So, in a sense, is that since it's not novel, you kind of have already a existing direction to know what this drug may do. Therefore, you can narrow your scope of research on toxicity related to EGFR and MET inhibition.
Thank you, Professor. And how do you balance the needs for innovation with the needs for safety in drug development for lung cancer?
I don't think it's a word of balance. This is not a fair word to use in this situation. Innovation is innovation, depending on how innovative your concept is. If this total new concept, then you may have to start from scratch to understand the toxicity profile in terms of the mechanism of action. But if it's not novel, it's just modification of the existing knowledge of what is already been there, that you can go according to the direction for what is known. So I don't think it's a matter of so-called balance. It's a matter of know-how.
So moving on to the next question. As we know, monoclonal antibodies cannot penetrate the blood-brain barrier because it is too large and so hydrophilic. Do you think, is there any possible therapy or innovation idea to overcome the intracranial metastasis?
So first of all, it's not universally not able to penetrate. there are cases of the patient that on amivantamab with CNS response. So it's not so-called that are universally not penetrable. But then you can say that it's less likely to be penetrating. But then you have to ask the question, what exactly is a blood-brain barrier? You know, how does the role of a P-glycoprotein, because a lot of blood-brain barrier, so-called, is not really a barrier per se, it's rather a glycoprotein pump that pump out the small molecule drug. So in a sense, is that the size of molecule does matter, but that's not the only thing that prevents it from entering the blood-brain barrier. So I think, yes, there are research that is ongoing in this arena, but then, you know, it is not necessary to just say that all monoclonal antibody cannot penetrate the brain.
Thank you, Professor. And at last, we would like to gain some insight from your perspective on the future lung cancer treatment. So, Professor Ma, in your opinion, how do you see the challenges of lung cancer research and the drug development being addressed in the future?
Well, I think that's a broad question. I mean, understanding that you're not medical background, but you are a science background. So you have to know what is there already first. Because if you don't know what is here, then you will not know what will be in the future. So right now, we have already made a whole arena of digital targeted therapy according to the so-called driver oncogene. And so this is an area of active research in the past 2 decades, and we have some good achievement. And then the other so-called wave of treatment that we have is so-called the ICI, immune checkpoint inhibitor, which is target mostly the PD-L1, and that also make a huge success in the treatment of the patient. Now, for the bispecific antibody is also under good research, but the concept is in a way similar to the PD-L1. You know, I mean, right now, one of the popular thing is a bispecific on CTLA-4 and PD-L1. Another ongoing active with some success is ADC, antibody drug conjugate, a monoclonal antibody with the cytotoxic drug on the side. And namely, there is now anti-HER2 ADC, anti-HER3 ADC, anti-MAT ADC, and all those are under very active research at this moment. So those are the things that is already here and ongoing. But in the future, I think we're looking in the potential of a cancer vaccine. With the success of mRNA for the COVID, I think mRNA technology may be used for cancer-specific vaccine. Cellular therapy is another one. And of course, the CAR T cell is already successful in lymphoma and leukemia. Certainly, we may be able to apply that in lung cancer, but you know, it takes a long way to go. And then the other area is the potential of drug edit and gene editing. With the CRISPR technique, we may be able to alter some of the gene. For example, people are looking at the potential to alter TP53, and also that, you know, by blocking out some of the so-called the driver gene where they can change the cancer behavior. So overall, it's very exciting. I think that all the new technology platform potentially may be applicable to the treatment of lung cancer.
Thank you, Professor. And then Professor Ma is an expert. Do you have any advice to our young researchers like us who are working on the developing novel therapies for lung cancer?
Right. I don't think that advice should be just for lung cancer, but just our research in general. So I do see that you come as a team, which is good. So I think for any research, it has to be a team effort. And also, I think if you are working in the laboratory, you may want to get attached or to get associated with some clinician, because right now, for example, you have your bispecific antibody, but you have to start asking questions, what would be the clinical application? But without the clinical knowledge, it's hard for you to set a direction to say what will be the future so-called implication of your drug is. So start to partner with some young doctors together with your development may help you to have the clinical insight on what is the need that's required in your laboratory research. That's number one. And number two, you have to actually acquire both the laboratory skill and also a little bit business skill. So for this generation, I don't think the pure scientist and discovery is a so-called that is a pure academic. But if you really want to transform your technology into a commercial product, you may need the commercial information and ability as well. So may as well in your learning phase, You learn a little bit about business when you go to school. Take a couple of business course, accounting course at least, so that you know a little bit how you may become an entrepreneur in the future. And that, last but not the least, you also need a bit of luck. That's the method of life in science as well.
Okay, thank you, Professor Wong, and thank you for your advice. Your advice is very useful for us. And I believe all of us and our audience have gained valuable insight of the NCRC and the advanced treatments. Thank you for being our guest.
And that wraps up today's podcast episode on bispecific antibodies in lung cancer treatment. I think everyone has a clear idea about how to maintain the safety and effectiveness of the new drug. Also, if you are interested in working on developing novel therapies for lung cancer, I believe Professor Mok's advice has given you a deep inspiration.
At last, we hope you all enjoying this enlightening conversation between our curious students and the knowledgeable professor. Please stay tuned for more exciting topics in the world of science and technology. Until next time, stay well, stay curious. Bye!